Okay, in the next segment, the last segment of the three-part set of online lectures on immunosuppression, we're going to wrap this up with some other ways that immunosuppression can occur naturally, as well as potentially through pathogens. Um, I want to say here that when we ended the last time, we talked about drugs that are immunosuppressive drugs, and they've been extremely critical to the ability to even do transplant therapy, and those drugs have made it absolutely possible. There have been other advances, of course, in terms of surgery, but for sure, drug therapies are in this case one of the most important ways. One of the most important advances in that field. Now, there are side effects of immunosuppression. It's not a benign thing. And the first, of course, is that when a person is immunosuppressed, they have an increased infection by organisms that are not normally pathogenic. So, cytomegalovirus and some fungi. Cytomegalovirus is one that is a real common thing. About 80% of the population um, is and has carries CMV. It doesn't cause you any harm. Your immune system keeps it in check. But if you become highly immune suppressed, this virus can pop up and cause some pretty severe illnesses. In fact, it can cause a form of blindness by attacking the eyes. And this is was well documented with for AIDS patients in the early days where we didn't have the drugs that we have for HIV. Um, that those patients got pretty nasty CMV infections in those days. We don't see that now because the HIV HIV drugs are so good. We don't see those. They're good at keeping the virus in check. The second uh, side effect is an increased incidence of some types of cancer, particularly lymphoma. So if you undergo immunosuppression, your immune system is also not suppressing or not observing or dealing with potential malignancies that may be occurring in your body. Now, they may be very small and insignificant at during that point, but over time, during if you have immunosuppression for long periods of time, this can lead to the development of cancers because they're just not being taken care of by the immune system. And in fact, in some cases, for people who have undergone immune suppression for transplantation, within a certain period of time, 5 to 10 years, they have much higher risk of developing those kinds of cancer. Of course, your choice may be dying immediately because you don't have a transplant, or living another 5 or 6 years with a risk of cancer. So that's the risk assessment that's generally that one might take. The major complication, though, that for sure is if major complication for sure is rejection. So this still occurs even with the immune therapy that we have, the immunosuppression therapy that we have, in a significant number of patients, particularly those in which the person is not a familiar donor. It's not a syngenetic match or a very good match. And even in kidneys, you still can see 25 to 30% rejection of kidneys. And we've been doing kidney transplantation for a long time. Um, so those are things that are that is one of the still the primary issue that you have to deal with, and it would be the one that would come the primary side effect that you deal with, and it would be the one that occurs the could be the most acute the most quickly. Of course, with For the first one, for pathogenic and for infections, you can treat those in many cases by antibiotic therapies. Um, Cancer therapy, of course, that's several years out before you would get that generally, and but rejection would be immediate. Now, congenital immunodeficiency, there are diseases uh, that we know of that decrease the immune system ability to fight disease and cause immunodeficiencies. And this generally occurs when you have one arm or one part of the immune system may be lost. Um, these diseases have been in some cases quite valuable because they're the term is they're referred to as experiments in nature, because if they're recognized by the medical community, they can help us learn about what happens when that part of the immune system is actually deleted or not part of the person. In most cases, a person can't live for very long without a significant part of the immune system. Most of the most of what we know about the immune system actually has originated from studies in mice, and mice are, for example, different than man, and in some, although there are some ways many may similar, but that difference, the degree that when we look at these therapies, that difference is not necessary. That the difference may not be readily apparent in those studies, animal studies relative to humans. So these kinds of experiments give us insight into that. These kinds of really unfortunate complications give us insights to the immune system. One of the first is Burton's disease. This is X-linked A gamma globulinemia, and it's X-linked, so um, it's a male. And so this involves Burton's disease, the inability to produce B lymphocytes, and therefore you can't produce antibody, and you have very low. There is a little bit of B cells there, but there's very little or no. It's so low, you get very little antibody that's produced. So you don't see that normal IgG production in the animal in the blood. Uh, there is normal. You don't know about this yet. There's no, but there are not really effects on delayed hypersensitivity, type 4 hypersensitivity, um, but there is a susceptibility to pyrogenic bacteria. Remember, IgGs and IgMs are really important for controlling bacterial infections. And you can treat this with gamma globulin injections. That means IgIV or IgIM, in which a patient is given antibodies that have been culled from several thousand volunteers from their plasma and given to the patient, and essentially it transfers the um, immunity of those thousand patients into the individual. Another one is DeGeorge syndrome, and this is called congenital thymic aplasia, and this has to do with no T cells, and in many cases this is worse because generally you get death from viral or protozoa infections. You can't give a person T cells from somebody else. Of course, they're going to react against that. They're going to react against that person, and actually go after her. And because the person's MHC1s are going to be recognized as foreign by those T cell receptors. So this is so loss of T cells is a very deadly disease referred to as DeGeorge syndrome. Uh, the last one is actually the loss of B and T cells, and this is due to congenital lack of adenosine deaminases or purine nucleoside phosphorylases. Um, Probably the first human disease which could maybe cured by human gene therapy because it results from the loss of adenosine deaminase. Um, Adenosine is actually quite toxic to cells if it builds up in cells, can cause trigger apoptosis, and so the loss of this enzyme causes in the B and T cells in the lymphocytes causes their results in their essentially their destruction. You can treat this to a certain extent by giving a patient adenosine deaminase that's pegylated. You can treat it by giving it, infusions of that. And for gene therapy, if you can stitch this back into the genomes of those cells, of cells, like for example, if you do it to the stem cells, if you could do that, bone marrow stem cells, then you'd be able to essentially cure this disease. All you need is because those that the enzyme would be expressed in those cells. Now, there's a famous case of and this involves David Vetter, who was referred to, as you might have seen, although this is quite some time ago, he was referred to as the Bubble Boy. Um, and he was born, it's a very tragic case, actually. He was born as a skid. He had severe combined immunodeficiency. That's what SCID stands for. Um, in which he couldn't produce B cells and T cells. And it turns out that his parents had a child, a boy. They had a boy. This particular SCID is also X-linked, and this particular congenital immunodeficiency is X-linked. And they had a boy who had died rather soon afterwards from infections, who had SCID. And it turns out it's a 50-50 chance. They had a daughter. She was fine. She did not have the disease. And then what happened is they decided to have another child. And there's some controversy about whether the doctors wanted them to have it or not. In those days, in late 1960s, we really didn't know as much as we know about the immune system now, and they thought that they might be able to somehow cure him if he had occurred. And plus, there's some controversy. There's some belief that the because this happened at Baylor at a research university, there's some, although this has been denied, belief that the doctors encouraged the mother and the father, the Vetters, to have another child. Regardless of whether that's true or not, they decided to have another child, take their chances, that 50-50 chances. Unfortunately, the coin that they flipped landed again on the wrong was landed again on the wrong side, and David was indeed SCID. He had to be immediately put into a bubble. Well, he was never touched by human hands until shortly before he died, and with great expense. And he lived in his until he was about, I believe, about 11 years old in the bubble. They fashioned a suit for him that he could get into, and it would take. Actually, this suit was designed by NASA. It took 24 hours to decontaminate the suit, to get it ready for him to be put on, and they could take him outside with with something that would control the air. But he grew up basically from a little tiny baby all out into 11 in this very confined space, and he also so there's a lot of questions as to where that was a really good thing. That was a good thing to do because, but in any case, he was a wonderful child and dealt with this difficult situation quite pretty well. And then around, he's getting bigger. He's getting to be close to being a teenager. Uh, the doctors felt like they may be able to control the skid and use sort of newer methods, though it's not clear what, to control the potential the potential donor's insusceptibility, and treat him with the bone marrow, give him a bone marrow transplant from his sister. Now, initially that worked, but unfortunately, she had apparently at some point been infected with the Epstein-Barr virus, which is the virus that causes um, mononucleosis, and actually is a transforming virus that can transform B cells into lymphoma cells. And people who have had uh, mononucleosis and Epstein-Barr virus tend to have a greater susceptibility to that. But it turned out that there is still some live virus that apparently was undetectable in her bone marrow, and that escaped. He was immunosuppressed. It escaped. Of course, he didn't have any way of fighting it, and he died within a matter of days. He had tumors just spreading throughout his body. It transformed the B cells, and tragically uh, died of lymphoma. Um, and he only was able to his mother, I believe. He only felt was able to actually touch him, with her, actually touch him without a suit, touch it, actually touch his body with her hands. Actually, during while he was in the process of dying from this treatment. Now, at that time, we didn't. It wasn't clear. We didn't know how to do these as well, and now this skid is treatable with the proper immunosuppressive drugs, and all. It's treatable also by very early if the neonate or the baby, particularly if it's a fetus, or when it's very early, if the child is given a bone marrow transplant, that can generally cure it. If it's not recognized past that, then it's potentially treatable by using enzyme replacement therapy with adenosine deaminase. And so advances in immunosuppressant therapy and how we do that, and how we now approach these diseases, have made this a manageable, potentially a manageable condition. Now you can have immunosuppression by other by the by pathogens. Um, in particular, the most famous one is HIV, and this is acquired immunodeficiency disease caused by the AIDS virus, or HIV. Uh, just to orient you for the virus is an envelope virus, which means it has a membrane. It's a membranous. A membrane that surrounds it. Um, it has the capsid encapsulates the nucleic acid, which is RNA. There is a reverse transcriptase there to copy the RNA into DNA. There's an integrase to take it into the genome. A protease, which is taken up, is can splice the polypeptide chain into pieces, and it has binding sites, receptors on its surface, GP120, GP40. And here's an electron micrograph, and you can actually see the capsid, and the little on the surface you can see the little receptors for GP120, GP40. Now, the way this virus works is it binds to CD4, and it also binds to a chemokine receptor, which is a co-receptor, and then is internalized, goes through an internalization process that's actually quite amazingly clever, diabolically clever, in getting into across the cell membrane, into the cytoplasm, where it uncoats, and the viral RNA is released, along with its reverse transcriptase, which is usually bound. That's converted, the HIV reverse transcriptase is converted into DNA, genomic DNA, that then is transported into the nucleus, and placed into and genomically integrated into the chromosomal DNA. That means when you are infected with a virus, it gets in the genome, and if those cells replicate, then you can't really cure a person, because the T cells that replicate, um, remember, make memory cells, which are very, very long lived. They can live for decades, right? We learned this earlier in the course. And so it's actually quite difficult to remove any of these virally infected cells because although T cells, normal T cells definitely have a certain lifetime, days to weeks, the memory cells can live for decades. So once it's integrated, uh, if in the presence, it turns out if you activate T cells, these CD4 cells with IL-2, or they become activated, you can get transcription of the, shoot, sorry about that. I don't know why I did that. You can have transcription uh, of that genomic viral DNA into genomic RNA, mRNA, that is, of course, exported into the cytoplasm and undergoes translation, ribosomal translation. There's viral proteins that are produced, capsid is formed, and then it's extruded through the membrane to make, and picks up its receptor molecules, GP120 and GP40, or then, as I say, goes through the membrane, are excreted in it, and they're placed on its surface. So those are the areas that, and so this is how the virus replicates, and it only replicates in activated cells. You have to activate those cells. And it just so happens that these 40, remember, you know enough now that if this results in the death of these cells, that how terrible that would be, because that would lead to quite a bit of loss of the immune response. Particularly, you couldn't form an AMI response and you couldn't form a CMI response. So you can one of the responses the CTLs do is these T cells will produce MHC1, right? They're nucleated cells. They'll produce MHC1. A virus a virus is growing inside this is growing in replicates inside the cell. Peptides are produced by that, so you can have MHC1 presentation of HIV viral peptides, which can lead to the CTLs destroying the virally infected cells, which are T cells. You can have ADCC, right? You can have their antibodies. You actually do produce antibodies. It's not true that your immune system doesn't recognize HIV. It does. It just gets overwhelmed by it, and antibodies can recognize the virus, um, recognize these viral receptors on the surface of cells that have been expressed and put on the surface of cells, and so that might um, if antibodies are there, phagocytic cells will then could attack these cells. And you can also have the binding of GP120 or these CD4 positive cells to CD4 positive. That should be 4 CD4 positive cells. And that would form giant cell, what are called giant cell fusions or syncytia. What that means is that uh, these GP120s right here can bind to CD40 on yet another T cell, and so they can fuse. An infected cell could fuse to a normal cell and make a giant cell, which has these two. The cell doesn't live for very long, but it has two nucleus. It's a mess actually, and so that can lead to the death of the cells. So cells. So how could affected cells, T cells, be killed? They could be killed through cytotoxic CTL response to the MHC1 response to viral infected cell. ADCC could occur, as well as binding, um, fusion of basically the GP120, allowing it to fuse to another T cell. In theory, you could also have potentially complement activation if antibodies bound to the CD120 and CD40 on those cells. So all those things can lead to the body destroying those infected cells, which would cause a drop in the amount of T cells, and therefore, if you have a drop in the amount of T cells, because your CTLs are killing them off, or your ADCs killing them off, or complements killing them off, then you become immunocompromised because you've lost these critical cells. So we see that here. If we can see in this figure, this is the number on the y-axis is the number of CD4 positive T cells. And so if a person has been infected, they become infected, they get sort of flu-like disease. It means they get sick. And what happens is you get an inflammatory response. The amount of CD4 T cells starts to drop. And at this point, you're starting to get 2 to 6 weeks in antibody conversion. So you're getting antibodies produced. So there's converted. And then what happens is that there's like a bounce back, right? It bounces back a bit. So there's an initial control of the disease. It bounces back a bit. But there's this long progression over time, though, of which your CD40s or your CD4 T cells just continually and continually drop. So that you sort of get an increase in the amount of the disease, an increase in the amount of disease. And this can take anywhere from 2 to 10 years. Once a patient drops below 200, that's sort of the magical level, that's 200 cells per microliter plasma, then they are essentially become so immunodeficient that they become susceptible to opportunistic infections. And that is why AIDS patients die if they aren't treated, because they die from these opportunistic infections. They also develop rare cancers because their immunosuppression can't suppress the growth of cancer. Kaposi sarcoma, for example, was in the early days. AIDS patients had that. They had CMV infections. They had just a terrible number of terrible what I would call side effect opportunistic diseases from the loss of their immune system. Again, we don't see that today because antiviral therapy is so good at controlling the virus that you don't, and therefore boosting the T cells, that you don't see these complications. You do see them in countries in which there is very little drug therapy, though. If we look at the response, you actually do the immune response. If we were to look at this over a set of days and then look at the antibodies that are produced, you actually do see in the first couple of months antibodies. You see antibodies start to be produced. So antibodies to the envelope proteins. You see antibodies to the core protein. You do see them being produced. You also see the generation of T cells. There are T cells, there are specific CTLs that are specific for knocking out, for going after those infected cells. And here you can see the amount of virus initially then drops. And so your system does fight it. It just can't get rid of it totally. And so over the period of time of 2 to 12 years, as the number of the virus load can start to go up, antibodies to the core protein go down, and this basically is this slow loss of, at this point here, you're talking about times when have gotten a fairly large loss of the T cells, and you get viral replication starts to increase. And in this case, this is then the time where you actually have what we refer to as AIDS. So those things do happen. Your immune system does take control of the situation initially. It just can't completely get rid of it. And the virus itself is actually in a number of different types. So it's almost a quasi-species. So a person who's infected with HIV has many different variants that they're carrying around with them. And those variants can not bind to antibody. They can, for example, is one of the biggest mechanisms of them avoiding being able, mutating so that they don't bind to those antibodies. So, so these are some of the mechanisms by which T cells are killed by the virus. One, you could have due to massive increase of the virions budding out of the cell, it would kill the cell. Two, virus helpers, which these T cells produce the GP120, um, and this can bind to the CD40 on another T helper cell, leading to cell fusion. And these multinucleated syncytia, they cannot survive and do not function as T cells. The virus GP120 can infect cells, it can elicit a CMI response, and so the T killer cells, the CTLs, will destroy those infected T helper cells. It's another way. Um, Lastly, viruses can produce excess viral components when replicated, and they get released when infected cells die. And so the circulating GP120, if you think about it, can bind to CD40 on uninfected cells, making them look to the immune system like they're infected, and they can then be attacked and killed. So you might see this would bind to CD40, and then it would track an antibody, right? It would track the CD40 binds to the CD4 binds to the GP120, attracts an antibody to bind there, and then you've got potential for ADCC and complement. So these are sort of the four mechanisms that have been thought of and investigated as why AIDS or why HIV leads to T cell death. So that's the end of this section on immunosuppression, which is incredibly important for having any kind of transplantation as even being a medical discipline. Again, the take-home messages that we start out with uh, are that that MHCs are HLAs are key determinants of organ donor susceptibility. Two, effective transplant therapy depends on understanding the reasons for immune rejection and, if necessary, suppressing this reaction. Three, natural immunosuppression can result from genetic defects affecting the function of those immune cells. We've seen that today. And fourth, pathogens can induce immunosuppression, and we've seen that with HIV. So that's the end of this section, and I will close here. And this is really the end of the section, the amount of the material that you will need to know for the exam.